Merci, Monsieur Plante, pour la présentation. Merci aux organisateurs pour l'invitation. Merci à André Gosselin pour l'estime que vous me portez. Aujourd'hui, je vais essayer d'accomplir une tâche très compliquée qui consiste à expliquer la pharmacologie des cannabinoïdes. Et donc, j'ai les lumières dans ma face. Heureusement, je ne pourrai pas voir votre face quand je parlerai. Je ferai mon mieux, mais pour le faire, malheureusement, je dois passer en anglais parce que des fois, même en anglais, c'est compliqué pour moi. Et donc, I'm going to do it in English. Also, it takes less time for your sake. Some disclaimers to begin with, and this is the outline of my presentation. So I will briefly tell you, because you definitely know already, how we discovered the endocannabinoid system when looking for a mechanism of action of THC. In fact, I played a very little role of it. This was mostly done by the group of Raphael Meshulam in Israel. And I will tell you that there is, in fact, not just THC. In fact, there is not just one cannabis plant. As you've seen, there is many cannabis plants, and their pharmacological use depends on their composition in these many cannabinoids or phytocannabinoids. And I will introduce the concept of the endocannabinoidome, which is an expansion of the endocannabinoid system. We'll tell you how, unlike THC, most phytocannabinoids interact with the endocannabinoidome rather than just the endocannabinoid system. And I will basically, this is one of the major messages I will try to do, that unlike THC, most phytocannabinoids have several pharmacological targets, and this is what makes them unique, and this is what probably makes them more usable in therapy and in the clinic than THC, whose side effects, which are the basis for its recreational activity, can be of hindrance and can be a problem in the clinic. And of course, I'll try to tell you if time allows, how we're trying to find an explanation as to why so many different plant cannabinoids with different chemical structures very often produce very similar therapeutical actions. So unifying theories. And I will conclude by basically telling you that there is a lot of hope that many of these compounds, beyond THC and even beyond cannabidiol, can be used in the clinic. So we all know how the endocannabinoid system was discovered. Basically, the history is a bit like the history of opioids and endorphins. And this is basically how the endocannabinoid system, the cannabinoid receptors first were discovered by looking for a mechanism of action of THC, and how, being their receptors for these molecules, people thought that there must have been also endogenous signals for these receptors, and therefore we named them endocannabinoids to distinguish them from THC, because they have a different chemical structures, even though they are capable of binding to the same receptors. And if I'm not mistaken, this is the only picture you will see of the cannabis plant in my presentation, which would be focused mostly, if not uniquely, on the cannabinoids, purified cannabinoids, whose pharmacology is already complicated enough. Just imagine if we wanted to look for the pharmacology of a cannabis extract. So the endocannabinoid system, I will make a very long story short, is composed of two major endogenous signals that are derived from arachidonic acid, anandamide, and 2-arachidonyl glycerol. They are produced on demand locally, so they're not hormones. They only act on neighboring cells or on the same cells whereby they are produced from phospholipid precursors. So their amounts depend very much not only on the activities of the enzymes that you see here, but also on how much arachidonic acid is present on the phospholipids that acts as the ultimate biosynthetic precursors for anandamide. And usually it takes an increase of intracellular calcium in the cell to stimulate these biosynthetic enzymes, to stimulate this phospholipid remodeling in a way that it can go to produce anandamide and 2-AG. And then once they've acted locally, as I said, they are immediately metabolized through enzymatic hydrolysis to molecules that are not active any longer on cannabinoid receptors. So these two compounds bind both to CB1 and CB2 receptors, which, as you've seen in the previous slides, are the two major receptors for endocannabinoids. There are two G protein coupled receptors. CB1 is very abundant in the brain and is responsible for the psychotropic euphoric activity of THC. Whereas CB2 receptors is very abundant in immune cells and is responsible for the immune modulatory actions of THC. What is this system therefore? After many, many years of studies, we all converged in concluding that the endocannabinoid system is an on-demand pro-homeostatic system, meaning that it is activated following a perturbation of the cellular homeostasis to try to restore the homeostasis of the cell. And in a whole organism, basically this means that this system can convey many important signals that are necessary for stress recovery and adaptation. And you see them here. These activities of the endocannabinoids are actually also activity for THC, but not for many of the other cannabinoids in the plant. So it is a beneficial system. It's there to help our cells, our tissues, ourselves to readapt following a stressful situation. But it's doing us well only when it is activated in a site and time-dependent manner. When it becomes dysregulated, this system can actually participate in many pathological conditions. It's a bit like the immune system. In order to help us, it needs to be well-regulated. When it's misregulated, it causes problems. And as you know, CB1 receptors, for example, have been involved in inflammation or in obesity or in the consequences of obesity. So based on these concepts, when the endocannabinoid system was discovered, there was a lot of hope that we could exploit this knowledge to develop new molecules. And of course, the first one that were developed were molecules similar to THC, because it was at that time already thought that activating CB1 could be a very good way to produce therapeutic benefit. However, as I said before, CB1 receptors are the most abundant G protein coupled receptors in the brain, and in order to preserve the beneficial role, the pro-homeostatic role of the endocannabinoid system, you cannot activate these receptors regardless of where they are. You can have different opposing actions if you activate CB1 receptors in a population of neurons rather than in another one. So when you take THC, you can have some beneficial actions, but you can also have some unwanted side effects. And the window between, the so-called therapeutic window, between the beneficial actions and the side effects is very, very narrow for THC. And the same when you antagonize CB1 receptors, for the same reason, because they're very abundant in the brain. This molecule here, you may have heard of, rimonabant, was on the market as an anti-obesity and anti-diabetic drug for 2 years. It was very effective. But in a few, nevertheless significant population of obese and type 2 diabetes patients, it also caused suicidal events and depression, anxiety. This is because CB1 receptors are important in the brain. You cannot just block them like that. So because of these reasons, people thought that we could actually exploit the benefit of the endocannabinoid system by indirectly activating this system, by using inhibitors of the degradation of the endocannabinoids, anandamide and 2-AG. And there is still hope that this can be done. However, so far, the clinical trials with the inhibitors of the enzymes that degrade the endocannabinoids, Pha and MAG lipase, have not been successful yet in clinical development. And I will tell you in a while what is one possible reason of this. But in principle, you know, it's a bit like using Prozac. You want to preserve the beneficial actions of serotonin only where serotonin is acting. If you inhibit the degradation of the endocannabinoids, you will preserve their site and time-dependent selectivity of action, of pro-homeostatic action, as opposed to what you do when you activate or inactivate their receptors indiscriminately everywhere in our body. So this was THC, but then people started to realize that there are there is a wealth of other compounds in the cannabis plant which could be pharmacologically active, and their activity was not studied yet. And you know, everybody knows cannabidiol, but also cannabigerol, cannabidivarin, tetrahydrocannabidivarin. For each of these compounds, in fact, there are several varieties in the plants. There is also the well-known fact that these compounds are not found in the plant as such, but they are produced by the plant as their carboxy derivatives. In fact, in order to get THC and the psychotropicity of THC, you need to heat or you need to desiccate for a long time the flowers of those cannabis varieties that produce a lot of THC acid. So the acid is not psychotropic, but when the molecules lose the carboxylic acid, they become capable of activating CB1 and CB receptors. This is true for THC, but not for these other compounds. These other compounds are also made by the plant as carboxylic acid. They can be decarboxylated through heating and desiccation to other compounds, to the compounds I showed you before, but they're not acting on, in a significant manner on cannabinoid receptors like THC. And like these compounds, there is many others. In fact, there is over 100 phytocannabinoids now identified. Some of them are present in trace amounts. Some of them can be more abundant dependent on the culturing conditions of the cannabis plants, the genetics of the cannabis plant. So there is a lot of biodiversity and therefore also pharmacological diversity in the cannabis plant. So I will go very rapidly to this list of biological activities just to give you the first message. Most, if not all, plant cannabinoids, except for THC, are not really capable of kicking very much the endocannabinoid system. Most of them can mildly modulate the activity of CB1 or CB2 receptors, the activity of the enzymes. You see here in blue, those compounds that can potentiate in a direct or indirect manner the endocannabinoid system, those cannabinoids that instead can mildly inhibit the activity of the endocannabinoid system, either because they act as inhibitors of the biosynthetic enzymes for the endocannabinoids, or as antagonists, or as allosteric modulators for CB1 receptors. So most cannabis-derived cannabinoids only mildly affect the traditional endocannabinoid system. It's a mild action, but this doesn't mean that it's not important. In order to understand better the pharmacology of the plant cannabinoids, we need to understand that the endocannabinoid system is more complicated than what we thought until the beginning of this century. In fact, along with anandamide and 2-AG, the two major endocannabinoids, there come a series of congeners. In the case of anandamides, N-acyl ethanolamides. In the case of 2-AG, the monoacyl glycerols. And there is other derivatives of long-chain fatty acids, like the acylated neurotransmitters, the acylated amino acids. We can calculate up to 200, 250 such compounds. So we went from 2 endocannabinoids to over 200 endocannabinoid-like compounds. These compounds do not, in most cases, activate CB1 and CB2 receptors. So they're a bit like the other phytocannabinoids I showed you. They activate other receptors. And you see here, in this vertical line, the receptors that have been identified for these endocannabinoid-like molecules. CB1 and CB2 is there, but there is also other G protein coupled receptors, ion channels, nuclear receptors, and so on and so forth. And there is a plethora of biosynthetic and degradation enzymes for these endocannabinoid-like molecules. Some of them are shared with anandamide and 2-AG, but some others are not. And this is why we now abuse of this OM term, and we refer to this expanded endocannabinoid system as the endocannabinoidome. And why are we interested in this? You see here another way of representing the endocannabinoidome, a list of mediators that are related chemically to the endocannabinoids, how they activate or inactivate some of those receptors I showed you before. The number of these receptors actually is increasing by the day. These are very promiscuous endogenous signals. You see here that the endocannabinoid system is included. It's not separate. It's different, but it's included in the big system of the endocannabinoidome. But we're interested in this because While the endocannabinoid system explains only the pharmacology of THC, the endocannabinoidome can explain the pharmacology of other non-euphoric cannabinoids. You see here how cannabidiol can activate or inhibit some of these endocannabinoidome receptors, like tetrahydrocannabivarin. And I mention these two because these are the two that have been tested in clinical trials along with THC. Incidentally, these endocannabinoidome receptors also act as receptors for endocannabinoid-like molecules coming from our additional organ, that is the gut microbiome. So, and I will come later to that, the endocannabinoidome can also be used to explain how the gut microbiome with its chemical signals can produce its well-known actions on our body. And this is to say that not all endocannabinoidome receptors do the same as CB1 and CB2 receptors. In fact, when it comes to the control of body weight, energy balance, inflammation, CB1 receptors does the opposite as most of the other endocannabinoidome receptors, and this is also true for some brain disorders, neurological or neuropsychiatric disorders. Even though in the brain, however, most of these receptors act in a beneficial manner. So let's go a little bit more into the detail. I don't want to bother you and bore you with too much pharmacology, too much biochemistry here, but it's important that you gather the message of the complexity of the pharmacology of these non-THC cannabinoids. First of all, the transient receptor potential channels. These are very nice, interesting channels. The first one to be discovered, TRPV1, was last year recognized with the Nobel Prize to its discover. It's not a G protein coupled receptor, it's a channel, it's a cation channel, and it's activated by capsaicin, but hot chili peppers, so other plants with potential therapeutic actions. Many other of these TRP channels can be activated by plants, some natural products with healthy applications, but mostly their role is to sense temperature. They can either sense high temperatures or low temperatures. They're important for us, for our fingers, for our mouth, to sense heat or cold. And it happens that we discovered that most of these receptors, they can actually be first activated and then desensitized by their ligands, in this case plant-derived molecules, but also by cannabinoids. Some of these thermosensitive TRP channels can also be activated by cannabinoids. In fact, while CB1 and CB2 are known as metabotropic receptors for THC, these molecules, which are also present in the plasma membrane, mostly of sensory neurons, are known as ionotropic cannabinoid receptors. And again, you see one of those boring lists, where you see a list of phytocannabinoids, how they can activate TRPV1. Most of them can activate TRPV1, even though cannabidiol is the most potent. Most of them can antagonize TRPM8, and this is a property that they have in common with the endocannabinoids. And basically, this knowledge is important because it allowed us to predict some potential clinical applications for cannabidiol, because we knew that some of these channels were very important for myoblast differentiation into myotube. This is a very important process that is always happening in our muscles when we're doing some exercise. So when we grow our muscles, well, not me, but most of you do, when you do exercise, you have some satellite cells that become myoblasts, and then myoblasts become myotubes. This process is very important. Also when you hurt and you have cut in your muscle, okay? It's a regeneration process that is very important. And it can be followed by measuring the expression of some biomarkers of myotubes. You see here myogenin, troponin. Okay, you see how short-term rather than long-term incubation of these myoblasts can align myoblasts and make them a myotube. This is a fiber, a muscle fiber. And some of these cannabinoids, in particular cannabidiol and cannabidivarin, are very efficacious at promoting this process. Only when you do the incubation with the cells for a short while, because if the incubation is prolonged to 24 hours, you see that the effect is declining. And this suggests us that this effect is due to activation rather than desensitization of these channels by cannabidiol. Why do we believe that this is you see here the same thing happening in human satellite cells with the cannabidiol and cannabidivarin. You can induce the differentiation of myoblasts into myotubes. This is important, and this is an effect that is mediated by TRP channels, because if we incubate the cells with an antagonist of these TRP channels, in the case of murine mouse myoblasts, TRPV1, in the case of human satellite cells, TRPA1, you can block these beneficial actions of CBD and CBDV, and in the case of the human satellite cells, also THCV. So this is pharmacology. I don't really want to bother you with this. But this is to say that CBD, to some extent CBDV and THCV, by activating either TRPV1 or TRPA1 channels, can stimulate the formation of myotubes. And this is important. And we tested this effect in vivo in an animal model of the Duchenne muscular dystrophy, and we found it to be very effective. At least in the mouse model of Duchenne, cannabidiol can be both anti-inflammatory and produce a little bit of regeneration of muscle. But also in the most known actions of CBD, epilepsy, TRPV1 seems to be important, at least in this model where basically you give an electroshock to mice and you see what is the threshold for these mice to develop seizures. I know it doesn't sound very good. It's certainly not very good for mice, but it was done in the full respect of the ethics. And you see how the cannabidiol in blue basically increases the threshold. It makes the mice more resilient to the electroshock before they give seizures. But if you repeat this experiment in the TRPV1 knockout mouse, which has no TRPV1 channels, you lose this effect. So this anti-epileptic, anti-convulsive action of cannabidiol is blocked, at least in this model, is mostly mediated by activation of TRP channels. So TRP channels are very important targets for non-THC cannabinoids, but also PPARs. And remember, these receptors were all listed in the endocannabinoidome. So the endocannabinoid receptors, CB1 and CB2, are only targets for THC, but some of the endocannabinoidome receptors are targets for many more phytocannabinoids. This is why this is one of the reasons why we studied the endocannabinoidome. Here you see some studies that started with in silico prediction of the conformation that some non-euphoric cannabinoids could assume, and whether it fits in these docking experiments with the conformation of some peroxisome proliferator activated receptors, alpha and gamma, very important for metabolic disorders, but also for neuroinflammation. And we could actually prove that the prediction done in silico could also happen in vitro. So these compounds, cannabidiol, cannabigerol, but also the corresponding carboxylic acids, which are the ones you find in the plant, can act as dual PPAR alpha and gamma agonists. This could be important both for metabolic disorders and some neuroinflammatory disorders. So it's a mess. When you look at cannabidiol, it basically does almost everything that it has been tested on. And this makes our life quite difficult. We have to be very careful in confirming these results, but you see here an incomplete picture of what cannabidiol does. Another important action, the potentiation of adenosine signaling. Some mitochondrial channels can be inhibited by cannabidiol. More orphan G protein couple receptors can be inhibited by cannabidiol. And there are also receptors that do not belong to the endocannabinoidome, like the dopamine, the serotonin, the mu-opioid receptors, sodium channels, sigma-1 and glycine receptors. They have been shown, at least in vitro, to be activated or inhibited, modulated in general by cannabidiol. Now, these actions, most of these actions, including the actions at TRP channels, PPARs, are observed in the low micromolar low, middle, micromolar range of concentration. So they're not very powerful effects of cannabidiol. Cannabidiol, unlike THC, does nothing in a powerful way. THC is very powerful. It's a magic bullet at CB1 and CB2 receptors. Cannabidiol is very weak or weak to a plethora of receptors. And you see here the potential therapeutic implications of these interactions. Is it important, is it good that a compound, once they were known as dirty drugs, molecules that could do a lot of things? Aspirin is one of them. Aspirin was given a Nobel Prize because we thought it would inhibit cyclooxygenase 2, but we know now that it does many, many other things. Metformin. So there is a lot of drugs that we use and we take that are not magic bullets. So magic bullets, this is a magic bullet, very potent and very selective for a target. But we can have molecules that are acting in a milder way rather than in the nanomolar range, in the micromolar range on more targets. For multifactorial disorders, and most of our disorders are multifactorial disorders, or if they're not multifactorial at the beginning, they become multifactorial while the disease, the pathology progresses. It initially affects only one target, but then only one tissue, but then the body adapts and other tissues, other organs become involved. So most, I would say almost all pathological conditions are multifactorials. This means that there is not just one receptor, not just one enzyme, not just one protein that is involved. Maybe it's useful that we have a molecule that has a lot of action on several targets. Or if one target is responsible for the side effects of a molecule, we could devise molecules that activate other targets and inhibit that target so that we don't see the side effect. So, in general, if we can predict their mechanism of action, polypharmacological dirty drugs could be more efficacious than cannabinoids. And as far as we know to date in the field of cannabinoids, cannabidiol has definitely many more uses than THC in theory. But you can actually, you need to find a way of studying this polypharmacology of phytocannabinoids. So we thought we could devise a pharmacological fingerprint. Now, if you do this with the THC, you only see two peaks, CB1 and CB2. If you do this with cannabidiol, and this is an out-of-date slide, you see many, many targets that can be either activated, positive peaks, or inhibited, negative peaks. And the same for other compounds. You see how THC is very biased towards only CB1 and CB2. So can we exploit the pharmacological fingerprint of phytocannabinoids? And most importantly, can we predict if this can give us a therapeutic action without giving us too many side effects? And of course, we know that we need to do that because at least in preclinical studies, all the non-euphoric cannabinoids that we've studied produce beneficial, promising actions in many conditions, sometimes different compounds in the same condition. So if different compounds in the same condition produces a beneficial effect, it's because it's either acting on the same target, on only one target, but for chemically different compounds acting on the same target, that's different, or it's because it's acting on more targets that are involved in that pathological condition. So that's why we devised what we call the therapeutic handshake. This is very complicated. I'll try to explain it. In French, it would take 2 days. So what you could do, but even if I knew how to speak French, what you can do if you know a disorder, you know what's happening to the molecular targets that are important for that disorder, okay? So you have, for example, 5 molecular targets in 3 different tissues. You know that those 5 molecular targets either need to be activated, and you see the green part on the top of the bar, and should not be inhibited, so that's why you see the red part on that bar, or vice versa, you have targets that need to be inhibited, so you have the green down and the red outside, or you have targets that in a particular tissue are indifferent, so they can be either activated, inhibited, or nothing happens. So in order to have a therapeutic action, from the literature, you can know each of these targets, whether it needs to be activated or inhibited. On the other hand, so this is basically you can from the literature, you can devise, you can see, you can visualize what we called a pathological, an etiopathological fingerprint for each disorder. It's a simplification, of course. And I've already told you before, you have a pharmacological fingerprint for a compound, or if you mix two compounds, you can have a combination of this pharmacological fingerprint. So below, you see those same targets, one compound, what it's doing on those targets. For example, cannabidiol. It activates the first one, it does nothing on the second one, it inhibits the last one. So you can basically, and this picture is slightly different in different tissues because the same compound can activate the same receptors in different tissues in a slightly different way. Pharmacologists know this as biased signaling. So you can devise a plane by uniting all those dots, and you can try to fit that plane into the etiopathological fingerprint. If those dots happen to be either in the blue or in the green area, in most cases, you will have a prediction for safety and for efficacy. But if they hit the red areas, you will see that some of those targets are being hit that are not supposed to be hit. Okay? So how good is this handshake between the pharmacological fingerprint and the etiopathological fingerprint? You can predict it based on literature data for each single pathological condition. It's a simplification, of course. So you could do that, for example, we tested this idea to see whether cannabidiol and mesalazine could be useful for ulcerative colitis. There are fortunately lots of models where you can test this compound. Here you see a list for a single tissue, the colon, of all the targets that have been discovered that participate in inflammation in the colon. There is more than 30 targets. And you will see that cannabidiol, or even the combination of cannabidiol and mesalazine, is neutral to most of those targets, thanks God. But there are activations or inhibitions for some of those targets. If you fit the pharmacological fingerprint of the combination of cannabidiol and mesalazine with the etiopathological, with all those bars, you can see how the line fits in the green or the blue without touching the red bits. If it touches too many reds, it's either going to produce side effects or it's either going to be ineffective. In this case, the fit was quite good. So we tested the combination of maximal concentrations of mesalazine and cannabidiol, and indeed we found that two inactive concentrations of these compounds were almost as efficacious as two maximal concentrations of the single compounds. So in this case it worked, but it also worked with Sativex. You know Sativex and Abiximols in North America. It was the first cannabinoid-based approved drug in the world and in Canada. Was the first country. It's a combination of THC of cannabidiol. Why was it devised? It was devised because there were epidemiological data and also anecdotal data suggesting that cannabidiol could tame some of the psychotropic, euphoric, unwanted actions of THC. So if you give THC with cannabidiol, you can widen up the therapeutic window of THC. You can give higher doses of THC without producing side effects. You can exploit better the therapeutic actions of THC. In fact, this is what happened with Sativex. You can give at least 10 times more THC than you would be able to do by giving only THC. So cannabidiol is minimizing some of these effects. Why is it minimizing that? Because it's hitting other targets that are responsible for those side effects. You see here a few examples. So can we devise a therapeutic handshake for Sativex in multiple sclerosis spasticity, which is the main indication for Sativex? Yes, again, we take the three most important tissues that are involved in spasticity. We see the targets that are involved in determining spasticity. We calculate the pharmacological fingerprint of the combination of THC and CBD in a similar amount. Okay, so you get this nice yellow line. You fit this nice yellow line with the therapeutic, with the etiopathological fingerprint, and you see that most of the interactions happen in blue or green parts. So Sativex is tolerated and is effective in many patients with multiple sclerosis. Can you do the same thing with cancer pain? Yes, we did that with cancer pain too, even more complicated. There is a lot of targets involved in pain, lots of tissues involved in cancer pain. The situation here was less good with others, but still promising. So the clinical trials with Sativex in cancer pain, however, were not successful, both studies. In fact, both studies gave exactly the same results. But if you read these studies, you will see that Sativex was effective only in the North American patients against cancer pain, not in the European patients. So nobody knows why the primary endpoint was met in the North American hospitals and not in the European hospitals. As a matter of fact, Sativex is not indicated for cancer pain. And finally, can we do this with cannabidiol alone and Epidiolex? Here you see a list of targets that are involved in seizures. Is again those that need to be activated, those that need to be inhibited, those that at different doses are indifferent. You calculate the pharmacological fingerprint of cannabidiol with Keppra or with cannabidiol and valproate. And in both cases, you see that the fit is quite good. Okay, so I'm almost done. I don't know how I'm doing with time. Yes, still 10 minutes, right? The pharmacological fingerprint is useful, but we need, we did it basically by hand. It took us 4 months to calculate those 5 or 4 pharmacological fingerprints. You need to do it in a better way. But there are other ways that can explain why chemically different molecules can produce the same effects on inflammation, on cancer, sometimes also on neurological disorders, on immune disorders. You will see that in the next presentation. So we went for a further simplification. Let's think of the disorders. The disorders we get are either hallmarked by too much or too little of something. Of what? Too much cell proliferation, too little cell proliferation, too much cell activity, too little cell activity, too much immune response, too little immune response. From the preclinical studies, we actually realized that most cannabinoids can actually counteract both the too much and the too little for some reason, maybe because of their multi-target action. So the cannabinoids are multimodal. In each pathology, there is at least one of these modes: cell proliferation, cell activity, cell response to the immune response. Not only that, metabolism. There are major controllers of metabolism. Here you see them: mTOR for autophagy, mitochondrial function, formation of reactive oxygen species. These are again phenomena that can be either too much or either too little. In either case, they accompany disorders. So cannabinoids have, at least in vitro, at least in animal studies, this almost magical property of being able to counteract both the too much and the too little. So this probably explains why molecules with so different chemical structures can actually be beneficial in most pathological conditions. A typical example of the multimodal action of cannabidiol, for example, this very nice paper published in JCI by the group of Tamas Biro showed how cannabidiol can be useful as an anti-acne molecule. Why? Because it acts on the cell proliferation, it acts on metabolism by reducing the number of lipid formation, and it acts also as an anti-inflammatory compound. The 3 major modal components of acne, proliferation, metabolism, and inflammation, can be tamed by cannabidiol. And this is a nice example. And you will see here that this is not independent, of course, of the action of cannabidiol on its molecular targets, TRPV4, the adenosine signaling, and so on and so forth. But there is maybe another way that can explain why chemically different cannabinoids can produce similar beneficial actions, and this is their potential interaction for the gut microbiome. When we eat something, the first thing that is going to feel what we eat is the gut microbiome. When we take an oral medicine, the first thing that is going to change or change the medicine could be the gut microbiome. So, as with any other drug, as with any other medicine, the gut microbiome could play a major role. And indeed, there are several reasons why we believe that the gut microbiome can be influenced by the phytocannabinoids, we and others. First of all, if you do in vitro studies, the plant cannabinoids, even at very low concentration, we're talking about nanomolar or low micromolar concentration, can produce this effect here, bacteriostatic actions, actions against the formation of films, and so on and so on, anti-quorum sensing actions. These are very important to control the growth of bacteria. But there is also now an increasing number of in vivo studies showing that some of the well-known actions of, for example, of Sativex, here in an animal model of multiple sclerosis, or of THC in this other respiratory distress syndrome, and our study in combination with fish oil in a murine model of ulcerative colitis indicate that some of the beneficial actions of oral cannabinoids can be due to the modification of the gut microbiome. And there are two types of modification, of course, you can imagine. First of all, the molecule that you take orally can change the relative abundance of the taxa of the different species that make our gut microbiome, and therefore the capability of producing metabolites that act on our own receptors. But there is also the other way around. The molecule, and this has been shown, for example, for L-DOPA in Parkinson, can be modified, can be metabolized by the gut microbiome. There is now evidence that some drugs are in fact acting as pro-drugs, that the gut microbiome is changing to the active drug. And if you don't have that right microbiome, you're not responsive to that drug. So the gut microbiome could be well involved in the so-called responsiveness to different drugs. And in fact, we know now that for some cancer immunological drugs, having a given microbiome makes you more or less sensitive to the therapy. And this is also true for obesity, where if you block CB1 or you desensitize CB1, can, of course, reduce obesity. I told you about rimonabant, but also chronic THC use can produce the same effect. And it has been shown that this effect and the ensuing endotoxemia, the chronic low-grade inflammation that accompanies obesity, that is due to the prevalence of some gram-negative bacteria in your gut, can be counteracted by acting on by the drugs that act on the endocannabinoid system. Okay, so I think I finished. Few take-home messages. So this is always not in this audience because it would be offensive, but this is always the way I start this presentation. Cannabis is not just marijuana. There is several types of cannabis with different uses. The therapeutic use depend uniquely on the cannabinoid composition of these varieties. Of course, other molecules like the terpenes, the polyphenols can also play a role, but most of what we know about medicinal cannabis comes from what we know on the cannabinoids. We must not forget that out of more than 100 cannabinoids, only one acts on the endocannabinoid system, which is composed of 2 receptors. The phytocannabinoidome, on the other hand, acts mostly on the endocannabinoidome, so the expanded version of the endocannabinoid system, which we discovered from the endocannabinoid system, which we discovered from THC. But there are also other targets. And of course, when it comes with predicting the efficacy and the safety of multi-target molecules, it's always a headache. It's always complicated. That's why we need bioinformatic methods. The therapeutic handshake, it's a primordial, very primitive way of tackling this problem. But I'm sure that the bioinformatic people and the systems biology people can devise and help us understanding and predicting the pharmacology of phytocannabinoids. And of course, as I very briefly showed you, we should remember about the other phytocannabinoids. There is not just THC, there is not just CBD, but there is many others that, like CBD, could be important. Let's not forget, however, that the carboxylic acids, the ones that you actually find in the plant, are also very promising, but they are complicated, because if you leave THC acid or CBD acid on the bench, on top of your shelves for a while, it will become slowly THC or CBD, so it will change its chemical structure because of the decarboxylation, and it will change its pharmacological activities and therapeutic benefits. So those of you who are thinking to develop these important compounds must find a way of making them stable, and this can be a headache. Those of you who are just using flowers of cannabis, remember that you may be wasting your time, because your flowers, if they're not properly desiccated, will contain increasing concentration with time of THC and decreasing concentration of THC acid, and the same for CBD, and the same for CBG, and the same for all the others. So it's important, and also important is to try that we start working on seeing the effect of these compounds on the gut microbiota. The gut microbiota has been involved in basically all mechanisms. Like the cannabinoids, it seems to be doing everything and the opposite, but with some attention we can understand to what extent the pharmacology of cannabinoids is due to their capability, that is becoming more and more proved by the experiments, their capability of changing or being changed by the gut microbiome. And just to thank the people who did the work, who are mostly in Italy. What I showed you today was mostly done in Italy. Here in Quebec, we work on the endocannabinoidome in relation with the gut microbiome, and maybe another time I will be telling you a bit more about this. But nevertheless, very great collaborators, despite the often opposite environmental and climatic conditions. Thank you very much.